Okay, I think we're live. Can everyone see me? Can everyone hear me? Are we doing good? Because yeah, there's been just technical difficulties all day. It has been a very frustrating day. Anyway, welcome to the show. We're gonna be doing something. We're gonna be doing something a little bit different today. As you can see, we are not in my kitchen. We're in the lab. Um, and a couple of streams ago, I don't remember if it was last week or the week before, we did a little fundraiser, and we for this thing. This is called a qubit fluorometer, and basically what this thing does is it has a little like LED light in it, and some filters and a basically like a tiny little light sensor. And basically the way this works is you make a dye solution. So it's like a fluorescent dye, but that dye only fluoresces, like it'll only glow if it's actually stuck to DNA, is the theory. So what this thing can do is you make your little dye solution, and the dye solution is always the same, and then you add a certain amount of DNA to it, and it tells you how much DNA is in the solution. This is really, really helpful because a lot of protocols, and this is actually something I've been struggling with, require a very, very specific amount of DNA or protein or RNA or whatever to work properly. It's like trying to bake a cake, but the flour is invisible, and if you use the wrong amount of flour, it just doesn't work. So this is this is theoretically a very, very helpful device. One tiny problem is that this thing is designed to use a very specific like dye set and controls. So like the company that makes this also sells the dye, right? And so it's kind of the one problem with this device is it's sort of the razor and blades strategy, which I mean I hate. And so I looked at the dye, and based on the way that it responds to light, the same dye that I normally use to stain DNA, and it also uses special tubes, not just special dye. It's special dye and special tubes. But yeah, so based on the specs of the thing, the dye that they use is approximately the same as the dye I have for staining gels. And the tubes that they use are just tubes. Like, the only thing that really makes them special is that they're, like, really thin and particularly clear. But, like, any tube should work. And if you calibrate around all of this stuff, you should be able to use just whatever. So there is one issue with this device, and I'm actually hoping that somebody here might know what's up and how to fix this. But we'll talk about that in a second. The other did we freeze? What's going on? Yeah. So anyway, we'll talk about the error with this in a second. The reason that the stream quality is a bit potato today is I ended up buying an HDMI capture card, but I'm a turnip, so I didn't read the ad properly, and so I got an outie, not an innie. So like the thing I bought lets you turn a USB port into an HDMI port, but only to connect to a monitor, not to like bring in video data. So whoops. So I got to return that and then get the proper one. So at the moment I'm just using my laptop. In future, I'll have this fixed so that we're actually using like HD cameras and stuff, so that like it's easier to see what's going on. Also, because I'm using my laptop, the angle is going to be a little shit today, and I'm sorry. But it is the way it is, and it's what I've got, and it's the only way we can get through the day today. So sorry. Anyway, so that's that. That'll be fixed for next time. As to the fluorometer, I did manage to get this fairly cheap, um, and so that's really nice. But it did come with a pretty big flaw. So I'm gonna plug this in, and I'm gonna show you how this this goes. So I'm gonna just boop to my other window so I can actually see what I see in live. Okay, so here's the device, right? So when I plug it in, so you'll see it boots and then it goes to the menu thing. Now, in theory, what's supposed to happen? So you'll see that it's got. See if I can get close. So it's got 4 different options on the thing, right? The DNA, the RNA, the protein are standard. Ion is a custom test, and part of the nice thing about this is the ability to theoretically upload custom tests. So that say I'm using a dye that isn't the thing that they sell, the machine can be calibrated accordingly, and then you can just use whatever dye you want instead of having to use their stuff. So let me show you the issue. Yeah, thing number 1, it should, if I put it, so it's got a little USB port, and if I plug the USB in, right, and then let's go to data. So you'll see, okay, you'll see the little USB symbol has a red thing next to it. So it's not actually recognizing the USB drive. But now if I do this, and I click on the power button, that turns the screen off, but but if I just press the screen again, it turns back on. Now if I go back to data, you'll see it's green, having changed nothing other than power cycling the screen. So that's really frustrating. And because of this, so the way that you put a new test in here is you have the test is actually on this USB drive. So in theory, when I unplug it and then plug it back in, it should boot and then immediately see the program, but it doesn't. It just boots. So I have no idea how to fix this. I've tried every formatting of USB drives I can think of. I've tried big USB drives. I've tried small USB drives. I've tried FAT32. I've tried FAT16. I have tried everything I can think of. I've updated the firmware, which was hard because I had to power cycle the screen to get it to read the thing. I don't know why this is broken. It's very frustrating. The problem is, the thing is, it does work as the device is intended, right? Which is cool. It does do the fluorometry thing, and we're going to do some of that in a minute. But what it does not do is let me put on custom tests. And so I actually managed to get a little bit of a refund from the seller, because this is a fairly big flaw. And I was like, hey, this is, I kind of bought this because of the custom assay thing, and, it doesn't work. And they're like, okay, well, I appreciate that. Here you go. And so that was really great. I really appreciated that. But now I'm stuck with a device that doesn't quite work the way that I want it to. So, yeah, that's kind of shitty. But anyways, if anyone knows how to fix this, I'm going to call customer support probably tomorrow. I was going to do it today, but stream. Then, if they don't have any ideas, it might just be, it could be, a friend suggested that it could be a bum capacitor. So basically I'm going to have to open it up, check every, power rail line and check for dips, and I'm like, I'm not an electronics guy. That's Chinese to me. I don't know what that is. I don't know how to do that, but we're gonna figure it out because this is a very cool thing and I want to make it work. But be that as it may, I did some preliminary tests with this, and whilst I can't do custom tests, and so because I can't do custom tests, I can't make this thing do the calculations for me. But the sensor works just fine. So I can do the calibration auth device. So like what I'm going to do today with you guys is we're going to make some DNA solutions, and we're going to make some dye solutions, and I'm going to make a spreadsheet. And we're going to basically just put in like known quantities of DNA at known quantities of dye, and then it should give me a nice logarithmic curve. And then from there, I should be able to be like, okay, cool. Now that I know how glowy each sample should be, I should be able to put in a test sample and then work backwards to figure out how much DNA is in there. And so this is my freezer right here. Just to show you, this is one box of DNA I have. I'll take the lid off so you can see this. It's a full box. Each one of these tubes has DNA solution in it, and I don't have any idea, other than the few that I got from like a synthesis company that like comes with a known quantity of DNA. I have no idea how much DNA is in each one of these. Like looking at it on a gel, I can do like a rough approximation, but like I don't actually know. So what we're gonna do is we're gonna make our little calibration thingamabooper and a huge amount of dye that's identical. And then we're going to actually go through and test some of these and see if we can figure out how much DNA is in them. Because that's actually really helpful to me. And then then the next stream that we do that's live lab work is going to be a lot of genetic engineering. So we're going to be doing a lot of transformations. So basically I've got two problems. Problem number one is I have no idea how much DNA I'm using, and so that's screwing with my calculations. Problem number two is some of the steps that involve putting DNA into... and also thank you to all the people who've been donating so far. That's greatly appreciated. I don't have a specific donation goal set today, because there's nothing I'm particularly looking to fund other than maybe buying some of the actual like assay kits that go with this machine, so that I don't need to do this like little home calibration one. So if you want to be pitching in and throwing in, it's greatly appreciated. But I don't have a specific goal today. And I guess the specific goal is food. I hear that's a thing you're supposed to do twice a day. Anyway, so problem number two, other than not knowing how much DNA I have, which is screwing with my calculations, problem number two is the step where you actually put the DNA into the organism. And so this is a problem both with yeast and bacteria, which are the two things that I work with. Is that for whatever reason, certain different strains of bacteria will behave differently when you go to do this protocol. And also different species behave differently, and there's different transformation protocols for each different species. So what I want to do, basically, is while just on a live chat, so that, we can hang out, I can do some work, you can ask some questions, and you can kind of like see my flow outside of a clean cut video. We're going to just do a ridiculous amount of transformations. Just ridiculous amounts. So probably like 10 or 20. I'm going to make a ton of plates. And we're going to just go through and fine-tune all of the reaction conditions and see what is the perfect protocol for me to get DNA into various organisms. And what's nice about this is basically once I have that really clean protocol, I'll be able to share it with all of you. So we'll be able to go and be like, okay, how long do we actually have to heat our bacteria up to make them work really well? Is it 30 seconds? Is it 45 seconds? Is it 90 seconds? Because I've been using 90 seconds on the advice from a friend, and that worked really well for a while, but now it doesn't work for a different type of bacteria, and I don't know why. So, gonna have to fine tune all of that. Anyway, are we lagging again? No, I mean we should be good right now. Not sure why it keeps lagging. Anyways, that is enough of me bumbling. This is very cold, so I'm going to put it away, because I also don't want it to melt, because DNA should not be heated and cooled repeatedly. Coolest of beans. Okay, so I'm going to just take 2 seconds before we get into this, and I want to read some of these comments, because we had some suggestions about the thing, and I want to see what people have been saying. Have you tried reformatting the USB? Yes, I've tried formatting the USB many times to every format I could think of. So that's not it. The only thing that should be on the USB, actually, and there's one other behavior that I can actually demonstrate to show you how weird this is. So this USB has a little LED on the back, so that when it connects, like when it's interacting with something, you can tell. So you can see as soon as I plug it in, it just turns on. So it gets power immediately, but it only actually, so I'm power cycling the screen right now. So you'll see as soon as I hit the off screen button, it starts to blink, and it, like, the data connection happens. And then when I turn it on, it actually can access the thing. But now I'm gonna unplug it and plug it back in, and you'll notice it doesn't do the flicker. Like, it's not sending data on boot. I don't know why. No idea. Could have been a brownout. Like, maybe it just doesn't have enough power when it boots because of a bum capacitor. No idea. So we've got a comment: adjusting the fluorometer for different dyes would require changing the excitation and emission wavelengths. So this device has 2 channels. It's got a green channel and a red channel, and there's like it's not like a very tight wavelength. It's a pretty wide emission and absorption filter. I don't know if those are, or excitation and emission filters. So as long as the dye is like within that range, it should work. And also most of the dyes that like glow greenish are all basically the same wavelength. It's like within a few nanometers here and there, and like the curve might not be exactly the same shape, but you can calibrate around that. So yeah, that's the thing. Okay. File name should be .qbt, drive should be FAT32. I've tried that. Didn't work. File. Don't forget about the file names. I've tried every combination of file name and everything else. Even if I, like, when I did the firmware thing, like the firmware update, it didn't read it at all. But because I can power cycle the screen and then hit a menu and then do the firmware update, that's fine. But because it only takes in new files on boot, and I don't know how to fix that. Use 1 GB drive. I tried that. Okay, so I'm done sitting cockeyed. So I'm gonna get comfy. Hopefully you guys can hear me reasonably well with me facing here and the mic over there. I'm gonna just open the chat really quickly on the other computer. I'm gonna reposition the camera, and then we're gonna actually get to doing some work, because I think I've talked enough, and this is meant to be a live lab work day. So we're gonna live up that lab work. Okay. Show your qubit file contents. Seems manufacturer induced. Yeah, probably. I mean, I had one. So I asked about this on Twitter, and I had one person comment and say that, like, you know, it only worked when you use the $70 1 GB flash drive that you buy from Thermo Fisher. And I'm like, I mean, at this point, I would pay $70 for the ridiculous fucking flash drive to make this work. But that's ridiculous, and I don't know how to do that. Anyways, I don't know how to completely reset it. If somebody knows that, like, if there's a way to, like, open this up and push, like, a hidden button or something, great. I have no idea how to reset it, though. Also, yeah, anyways. Blah. Okay. So, I have my commentes open, so I can actually see what y'all are saying. I'm just going to open up my Streamlabs tab so that I can see if people kick in and I can thank them appropriately. Which model is it? This is the Qubit 2.0 fluorometer from Invitrogen by Life Technologies. Connect through a powered hub. See, that one somebody suggested, like, I saw that suggestion, and that one actually seemed almost plausible. Like, because if it is a brownout, then maybe that fixes it. Like, maybe that would fix the slight lag. No idea. Okay, so I need to pop out the chat so I can open this at the same time. Okay. Cool. Also, for those of you who have been asking, like, when's the next like proper video coming out, that's what I'm working on the rest of my day today. We're doing a video on the lactase update, which actually, since I happen to have it handy, and literally right in front of me, which one's which? So on an earlier stream, I mentioned I was waiting for a solution to turn blue. Yeah, I think it turned blue. The tube on the darker tube, so the one on my left, but so this one's the control. So this one uses like commercially produced lactase, and then this used homegrown lactase, and you can see they both turned blue, which means there's lactase in both of them. So I have actually managed to grow some lactase, and you're going to learn how to grow some lactase in the next video. It's going to be exciting. I'm excited. It's going to be some fun. Okay, so I'm going to have to tip this down a little further, which, yeah. Again, apologies for the craptastic angle. It is the best we can do with our current equipment. What with the, I'm an idiot and I bought the wrong capture card. Okay, so these are the weird, special, magical tubes of magicalness. Kind of. These are not actually the ones that the company sells. These are instead ones that I found on Amazon that seem to satisfy the same thing. So these are what we're going to be using for measuring our samples. Now we need to make a lot of the dye solution so that we've got enough of it, basically, so that it's always calibrated. So for that, I'm gonna grab my little tube rack. I love these things. They're 3D printed tube racks. They're a lot of fun. Okay, so I need that. Need a tube. So, like I said, I'm going to be making a lot of dye because I don't want to have to do this again, basically. Okay, so I've got my, I keep these under the table. These are long serological pipettes. So these are really nice because they're graduated. I don't know if you can see that really well because, again, potato camera, which, again, I apologize for. But yeah, serological pipettes, which mean I don't need to sit here like a jackass pulling 1 mil at a time over and over again if I need 10 milliliters or something. Okay, where, now, big $1 million question, where is the thingy that this works with? Which I have evidently moved at some point setting this whole thing up. And I do not know where, or maybe I dropped it. What did I do? Where did it go? I don't see it. Well, this is exciting. Alright, well, I don't know what happened. Where is the thingy? Did it fall? Did I drop it? I don't know. Okay, where do I keep the other ones? Well, this is silly. This is like very, very silliness. Oh, I found it. It did fall. It's still that new lab-ish, you know what I mean? It's the yeah, that's ooh, somebody pitched in. It's in Russian. I can't read Russian, so I can't thank you properly. But thank you, internet Russian stranger person. Donations are greatly appreciated. Okie dokie. So, this doesn't actually need to be overly sterile. It's just, I mean, it kind of does, but like, we're not talking like surgical aseptic here. We're just, you know, don't lick the tubes. All right, come on. So just off camera, I've just got some freshly autoclaved, sterile water. Just regular water, nothing exciting. Not even buffered. We're just putting water in a tube. Oh, somebody kicked in. Okay, let me just do. Wonderful. That should be all of that we're gonna need to do today. So that's nice. Throw a lid on here. Answer some. I saw some stuff go by in the chat. Where is the new lab? Still around Jacksonville. I haven't lived in Jacksonville in, oh fuck, like 2 years, 2.5 years. And if I never have to go back, it'll be too soon. Wow, Florida is a weird, wild, and awful place. Like, not a fan. Anyway, okay, so I've got my little, this is my little container of, I try and keep all my stuff like self-contained into little containers. So this one is all of the stuff I use for like loading gels. So this way, every time I need to run a gel, instead of having to like reach into the fridge and like dig out, you know, 10 different tubes, I just grab my little box and we're instantly ready to go. But I'm gonna store it in the fridge until used. I gotta say, I love having this little mini fridge like right next to me. It's made this just an absolute pleasure. Because, you know, then I can just like reach in, grab stuff, which is great. Cool. So the last time I did this test, I made 400 microliters worth of my like test dye solution, and I used half a microliter of my actual dye. So that's 1 microliter for every 800 microliters. We're gonna call it 1.2 microliters of dye for every milliliter. So that means I need 12 microliters of dye for this 10 mils of water. So I'm going to use my P100 for that, which is this guy, which means I need my P100 tips, which are over here. And I am going to just use the cubit as my work surface, because as you can see, my work surface is extremely tiny because I'm trying to cram 2 laptops on here because I am a turnip. Okay, cool. Big lag? Are we lagging? Yeah, I don't know why my internet's lagging. I called and I yelled at the I say yelled. I I talked to tech support for my like my internet company, and their response was, "Shrug, we have no idea why it's so slow." Lag. Yes, I understand. Are we good? We should be back now. It's back. Okay. Again, sorry about the lag. Can't really do anything about that. Like I said, I called the company to ask what the deal is, but their response was shrug. To whoever said you need Starlink, how about fuck no? Starlink is just sky cancer. It's awful. Hate it. I mean, it's neat to take pictures of because you're like, ooh, that's a lot of satellites, except that it's going to start messing with people's astrophotography. And I love astrophotography because astrophotography is dope. Okay. Coolest of beans. We now have our dye solution, so that's real good. Now let's grab some of these babies. Where are my little tweezer things? Here they are. So, something I like to do, rather than reaching in my grubby hands and, like, getting, the enzymes that might be on my gloves in here, I always have a pair of really fine-tipped forceps. These happen to be titanium because I'm a huge fucking nerd, but not necessary. So yeah, this way I can just reach in, grab it by like a corner or something, somewhere not important, and then do that. I don't really want to waste a ton of these tubes, but I don't care, because if it means that we get... Well, that one's fucked. That one's fine. It means that... So I'm going to need probably 6. Because I want to make a really, like... The test that I did last time, the divisions of stuff I did wasn't really ideal. So I'm gonna grab 6. So we're gonna have one empty. We're gonna have one at 250 nanograms per milliliter. We're gonna have one at 500 nanograms per milliliter, and then 1,000, 2,000, 3,000, 4,000. Should do. And one's just gonna be dye as our like negative control. So we want a total of we want the same amount of dye solution in every single one. These tubes should be 0.5. Yeah, I think these are 0.5 milliliter tubes, which is the size that fits in the machine. I literally only use them for this. I'm going to set this to 190, because that should give us the ability to do basically everything that we need to do. Yeah, that should work. That'll give us 10 microliters worth of volume to play with, basically, which is, frankly, should be more than we'll ever need, because if you're assaying 10 microliters of something at once, you're really wasting a lot of reagent. Most of the time, if I've got a little tube of DNA, it's got 50 microliters in it. So to measure 10 microliters of 50, that's using a fifth of my stock at once, which is insane and stupid. Okay, so let's tip this a little further down. Again, which is going to be just a shit-tastic angle, but it is the way that it is. Also, I realized that this dye solution is actually warm because I just took the water out of the autoclave. And yes, I'm reusing my tip. That's fine because I'm just distributing, and everything should be sterile. Cool. Dye has been distributed. So now I'm going to just close the lids for all of these. For exactly this reason, so they don't get knocked over. Has the lag really been bad? Like it's hard for me to look at the thing and work at the same time. And because I really don't know what to do about it. Like I've called, I called the company, I upgraded my internet speed, but it's just been potato for like a week now. Okay, I'm gonna pull the DNA out. Not that bad. Lag is fine. Okay, cool. So I don't know how much DNA I actually have left in here, but there should be enough. I'm actually using the spider silk DNA as my control because I have so much of it. I got 10 tubes of 100 microgram thing. So I have an enormous amount of this DNA. Also, I figured out. I think I figured out why the spider silk hasn't been working, which was both gratifying to know why, but also very, very frustrating because it's not an easy fix. Well, I mean, it is an easy fix. It's just a slow fix because it'll take me a little while to replace. Because so basically the long and the short of it is to be able to get this spider silk DNA into yeast, you have to first cut the DNA with a restriction enzyme, so make it from a little circle into like a line of DNA. And the enzyme that you need for that I don't have. I've ordered it, but I don't have it currently. I didn't know this, and there was no documentation to tell me this. I had to like find this in a paper. But yeah, turns out you gotta cut the thing, then you transform it, and then it like magically works or whatever. But so that's super annoying. The company that made this though, I'm seeing if they'll just do the transformation for me, because then it's literally no work. It's like I have essentially paid them a fee, and they've not only made the DNA for me, they've also put it into the yeast for me. So they'll just send me the modified yeast, and then I'm done. Anyway. Okay, so I want to label these before I start putting DNA into them, because otherwise I'm going to get confused, and then we're all going to be sad because I'm not going to know how much is in here. So we're going to call that 0. We're going to call this 250, 500, 1,000, 2,000, and 3,000. So these should be fairly good. The one thing I am going to have to do is I need a little tube, like a little PCR tube would be good. Just because I need to dilute this a little. Okay, so I just grabbed a little PCR tube. What I'm going to do is I'm just going to dilute the DNA a little bit, because as it stands, it is just a whiff too concentrated, so it makes handling it kind of annoying. So like I can put it directly into here, 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 and here, but this one I'd need 0.25 microliters, which my pipette can't do. The lowest it can do is 0.5 microliters. So what I'm going to do is I'm going to take one microliter. Yeah, I'm gonna do one. That's really a tiny amount of DNA though, like a tiny amount of solution. It'll take 5. No, let's just do 1. All right. Well, let's do this then. Okay, so I've got my little bit of DNA solution in there. I need now a microliter of water. And then I can just take 1 mic of the result. That should be fine. Or, no, I take half a mic of the result because I need a quarter. Okay, we've got our tiny little bit of DNA in there with a tiny little bit of water. Now I'm going to take half a mic and load it into the next tube. Cool, we've got DNA in our first tube. Very exciting. Now let's put some DNA into the next tube. And this should hopefully give us a reasonable calibration. Yeah. So that way we can actually measure some of the other samples and see what's what. And see if all of our math checks out. Yeah, if it's alive, everything has DNA. Or RNA, I suppose. But other than viruses, everything just uses DNA. And then people get all weird about are viruses alive? Yes, viruses are definitely alive. I'm trying to work where you guys can see me, but again, or not again. This is biology. So everything is just moving small volumes of liquid around, so it doesn't really look like much until the end. And I've gotten some comments from people being I'm bored of the bio stuff. Go back to doing physics or chem or whatever. Apparently they don't realize that there's a frickin' pandemic, so my ability to do those other things is essentially non-existent. And as such, I am sort of forced to just do bio stuff all the time. Not that I'm complaining, because I am trained as a biologist. I love biology. Also, the things that I'm doing, as soon as they work, will be some of the coolest things I will have ever done. So, also it's my also as somebody else points out, it's my goddamn channel. I can do. Oh wait, I did not adjust that. That was stupid. It's my goddamn channel. I can do whatever the fuck I feel like, and no one can tell me otherwise. That is the whole point. And I intend on doing exactly that thing. And what I want to do is bio. So the next couple of videos that I've got planned. So the one that's coming out in the next couple of days is making lactase, talking about the lactase gene therapy, talk about how it went, talk about the plan moving forward. That'll be fun. Then the next video after that, and I don't know if this is going to be the exact order, but the next one that I'm working on is the beta-keratin yeast that I showed off in the last stream. In fact, I actually have that handy. So if we just, I'm going to tilt the camera up for a second. So do you see that flask right here? Let me get this out of the way. There's a flask right there with a little airlock on it. That is actually 500 milliliters of beta-keratin modified yeast. So the yeast produce beta-keratin. I might actually be able to just reach in and grab it if it's on the top. Yeah, here we are. This is the yeast I'm talking about. Oh, that's very drippy. Very drippy. Very bad. Drips are bad. Just dump out the extra water. Anyway. But yeah, so you'll see this is extremely orange. So these yeast have been modified to produce beta-keratin. So we're going to be talking about how I made this and making some. And so that's going to be really cool. And then the other thing that I've been working very, very diligently on is the spider silk yeast project, which has been kicking my ass because it's really hard. It is the hardest thing I have ever done. like hands down. I've done a lot of very, very hard things. I've built a nuclear fusion reactor at one point. That was orders of magnitude, like orders of magnitude higher, like easier than the spider silk is. Spider silk is basically I'm trying to force a yeast to produce a protein it does not want to make. Handling the DNA is awful. Like handling the yeast is awful. Making the two fit together is awful. It's just it's difficult and a pain in the ass the entire way down. Okay, we have all of our samples made, so now I can measure them. So I'm gonna just pop open this. Just pop open a thing and get a spreadsheet going. And I can't show you the spreadsheet because I'm doing this on Google Drive on another computer that you can't see. But I'll be able to tell you if it works. Yeah, I think the best way I've heard of describing the thing I do, like the genetic engineering stuff, is it's like trying to code, except there's no except the compiler is like actively hostile against you and wants totally the opposite thing that you want, and so will go out of its way to fuck up anything that you do. And so that's bio. It's the code is ridiculous, and you have to pay to have the code made, and the compiler hates you. And so you've got to like work through that whilst simultaneously trying to like do your shit, and it's just non-compos mentis. Okay, so let's start with our 0. Read AVU. That seems very high. Okay, I'm gonna just do a little quick testy test. And I want to just measure the big one and see if it's sufficiently different. It is not. This is... Is that leaking? Where did this water come from? Okay, I'm just doing a quick measure on some of these just to see how they're responding. Okay, so that says 3.6. 3653. This one says 5.5. Am I measuring it the wrong wavelength? No. I'm doing this right. I've got the same recipe of stuff that I used last time. Oh yeah, sorry, I forgot to tip you guys back down. This is very frustrating. So literally all I'm doing is here's the thing. I'm just putting in the sample. So this is supposed to be the empty blank one. And I click read. So it's reading out an arbitrary value of 55363. Okay, that's the empty one. And then if I put what should be the most concentrated one in, I'm gonna give a good mix. I click read. Oh, good, now it's working. Okay, so this was a thing that I found the last time I did this, which is if you don't mix the tubes sufficiently, and like it doesn't like the dye doesn't interact with the DNA properly, and then it doesn't glow properly. So now it's reading out 74799, which is actually expected. That's normal. I mean, it should be a higher number when there's DNA, whereas when compared to when there's no DNA. So the fact that it wasn't doing that was a little weird. We're just going to give these a good little mixy mix. And I'm going to take a minute or two to answer some questions while we let these just like sit and do their thing and figure out their life choices or whatever. Okay. Um, all right, cool. Well, let's give these a measure. Let's try this again and see what goes on. Theoretically, there could be an issue where um, basically every time you fire every time you turn a little light on, technically you break some of the dye. So the number could go down each round. Um, yeah. Jacob asks, why do you think a virus is alive? Because this is one of those, if it acts like it's alive and like, I mean, the only thing that makes a virus not alive is the fact that it requires other alive things to replicate. But like, um, I don't know if that's a fair reason. Like I don't know if that's a fair reason to say that something isn't alive. Um, five, 5, 4, 5, 4, 3. Because I mean, if just being a parasite makes you not alive, then there's a lot of very much alive things that we wouldn't count. And yes, there's other definitions where it's like, well, does it produce waste, and does it have cells and all these things. But it's like those like, who really cares about those definitions, if we're being honest? Um, like those were made up by people whose only tools were a microscope and didn't know their ass from a hole in the ground. So I don't know why we're taking their definitions as the be all and end all of like what is alive, especially in the era of like computer viruses, where they act very much alive. Oh, what the hell? Come on, this one is supposed to... That is supposed to be a higher number. Why is it not a higher number? Now it's a higher number. Okay, just needed a good mix. Fuck's sake. 2, 3, 1, 3. It's very weird. I mean, like, it really does need just a really, really good mix, seemingly, to get it to actually work. And yes, I realize this looks ridiculous, but it's the best way to shake the tube that isn't going to shake the entire table. Read, por favor. Also seems like any like bubbles can mess with it, or like if all the liquid isn't quite in the right spot. Yeah, okay. See, now this is the thing. It's like you really gotta maintain certain parameters, but then this works great. So I'm just gonna read off the numbers I've been getting. So for remember that the order is 0, 250, 500, 1,000, 2,000, 3,000. So for 0, I got 54543. For 250, I got 59165. For 500, I got 62313, and for 1,000, I got 68008. So like the number is going up, and it's doing so pretty evenly and consistently, ish. So I'm probably going to go back through and remeasure all of these again just to like see what is up. But it seems to be working. Like I seem to be getting a reasonable set of numbers. So I'm going to graph them in a second once I'm done getting all of these values, and it should, in theory, see like that is a perfectly wonderful functional number. 5, 2, 8, 8. I almost wonder if this is not logarithmic and if I was just measuring shit improperly last time. Again, we're gonna give this a good little mixy mix. And I'm just trying to standardize. So it's like, give it a mix, give it a shake, take the reading, see what it does. See, like that's weird. So this is either bottomed out or I fucked something up. Because now that number is smaller than the one that had more DNA, or had less DNA in it. So, not really sure what to do about that. we're gonna see. So I'm gonna graph these now, and we're gonna just see what at all this looks like. See if it makes sense. See if it looks good. I'm only gonna graph the first 5 because that last one was fucked. Okay, insert a chart. I realize I'm not really looking at the comments. I will look at the comments in just a moment. I don't want a column chart. I want a no. I don't want. I just want a line chart. Just want like a normal line chart. A1 to A5, okay, add another range, B1 to B5. Okay, let's see if that fixes it. Nope, that does not fix it at all. See, now these numbers actually look better, ish. I just need to adjust the thing. So I'm seeing a lot of like vortex to mix. Yeah, I know. Customize horizontal axis. I don't know why this is starting in such a ridiculous thing. What? What if I get rid of this? Click OK. Okay, I'm gonna start this again. Sorry about this. This is now just me failing here. I'm gonna turn this up because now you're just looking at me. I think your light transmission is bad with those tubes. Yeah, probably. They're not the tubes that I'm supposed to use. But I want to see how close of a thing we can get with just using cheap stuff. You know what I mean? Because I mean, I'm gonna buy the actual stuff, and like, if y'all want to kick in some donation money, I'm gonna help me along in the making of that thing, or the like buying of that kit. But I want to see what we can get done with just the stuff we have available, basically. You know what I mean? Like, because the kit is another like $300 or something for like 100 samples. It's like it's dye and water. So if I and their tubes are $100, which is insane. Which I just find that ridiculous. So let's data range, add another range. Okay, this is actually looking really quite good, I think. I'm using gel green. Is the dye I'm using? Vertical axis. Minimum value. We're gonna call that 54543. Okay. Okay, this is actually looking pretty good. This is looking pretty good. We have a curve. It's not perfect, but you know what? That is okay. Let's put a trend line on there. Logarithmic. No, it's not quite logarithmic. It's not quite linear. It's sort of moving average. That's sort of not helpful. But whatever. This is the way that it is. I'm using gel green, or maybe I'm using CyberSafe. I honestly can't remember. The tube says safe DNA gel stain. from Bioland Scientific. I have no idea. I will show the curve in just a second. We're getting there. Is there a safe way that I can open this without it being just ass cancer? Yes, actually, there is. Okay. Okay, so I'm gonna just do a little flippy-roo. So I'm gonna have to close some stuff really quick, just so I can just so I can open up this spreadsheet in Stream, like with the Streamlabs thing, so that way y'all can see what I'm seeing. So just take a moment. So just gotta close a bunch of other stuff. Okay, this should be safe enough to open. I'm just not sure why this isn't. Ah, there we go. Cool. Now it is. Okay, let's go to Streamlabs. That should do the thing. Wonderful. Okay, so now I'm just gonna nip over to here. Make sure that this is all looking real good, which it is. And okay, so you should be able to see the curve now. You'll see it's not quite linear, but that's fine. So I mean we do seem to have all the values we need. So we've got our 0 down here. We've got up to 2 micrograms per milliliter. So now I had a chart before, so I'm gonna see if I can find that. Where was it in my other drive? It was in my other drive. This is the problem with having like a bazillion different Gmail accounts. I think it's in here. Have a look at the thing. Plot it on a log scale. I could try and plot it on a log scale. There we go. Horizontal axis, log scale. Yeah, on a log scale it curves up, which is not really more helpful at all. Wait, did I? Oh, that's the horizontal axis. We want the vertical axis on a log scale. Log scale. I still don't think that makes that better. If anything, that makes that worse. Okay, well, meh. So now that we have our calibrated solutions, I want to try. I want to try a test. I want to try a test sample. Find the regression line. Is there an easy way to do that? I never use Excel. This is so not my thing. Series. Oh, okay. Polynomial curve seems to work really well. Show R squared? Is this the... No, that's not helpful. How do I... Okay, well, I don't know if this information helps at all, but, okay then. Okay, well, now that we have our chart, I'm going to set this back to the other view, and then we're going to try and measure an actual test sample. That sounds probably the best. And then we can see if any of this actually worked properly. So we're going to just do external cam. Boopity boop boop boo. Says to get a meaningful calibration curve, you need linearity. Yes, I'm aware. We're going to try this, and we're going to see what we get. So I'm going to... Where's my bag of tubes? bag of tubes. Right. Okay. I'm going to tip you guys down more. See the thing. Okay, just grabbing my DNA. Okay, let's pick a tube, shall we? I'm gonna pick. I don't know. I got a lot of tubes here. Oh, you know what? I'm gonna use this one. There's not a lot left, so we're only gonna use a microliter, and we're gonna see how much is in each microliter. Actually, you know what? Screw it. I'm gonna pull out a couple interesting ones. Ones that like I know that I've used. This one. Okay, so the ones that I'm pulling out are one that does. This is a GFP, so this is like an E. coli GFP. This is my favorite GFP. It's that ridiculously green one, and I love it. We're gonna pull out this RFP. We've got the beta keratin because I actually need that one for a thing, and we're gonna give this a try. We're gonna see what we get. And yes, I realize I could have been much more careful about how I built my curve, but that is okay. We're rolling with it. So we're going to do exactly the same thing. We're just going to pull out 190 mics, add it to my little tube. And then, which one should we try first, do you think? I think to be fair, I'm going to use... I don't know. Let's just try one microliter. Then we know it per microliter. Wow, the comment section really is just the worst today. Can y'all not? Okay, we're gonna start with number one. This is my GFP. So let's see how much DNA is in here. And again, I realize I am doing this the most difficult way possible, and that I really should just buy the kit, and I will buy the kit. But I thought this would be something that's just fun. Oh God. People were asking. People said I should vortex. So I'm gonna vortex. It's off camera. But that's what that buzzy noise is. So we're gonna let that just sit and cook for a minute. I'm gonna give it a label. Because remember, kids, it's always important to label your tubes. Because if you're an idiot and you drop them upside down like that, then you're very sad when you don't know what they are. Cool. That's going to just sit and cook for a bit. So did anyone else have any ideas about how to fix the qubit? Because if anyone had suggestions that are like actionable, that would be awesome. Thank you, James. Okay, so we're gonna just let's fire this and see what we get. A little nervous. Come on, a number that makes sense. That is not a number that makes sense. that is worse. That is a worse number than before. Okay, I'm going to try reading my 0 just to compare, because I remember it being higher than this. Yeah, that doesn't make any sense at all. How? What? This says there's negative DNA. That is not helpful. That is not helpful at all. What the hell? There's the same amount of liquid in each of... Is there the same amount of liquid in each of these? Yeah, it looks like it. Well, this is just agonizingly stupid. Meh. I think this is very quickly turning into the... I'm going to just have to buy the kit. I just wanted to try this just because I thought it would be fun. Wow, that is literally negative by like a lot. Okay, I think that might be a wash then. Let's try this. We're gonna try this one more time. We're gonna spare one more tube, and we're gonna go from there, because this is so far this is not working. So let's just start this again. We're going to use a different one of these. We're going to say maybe the Keratin one. I mean, it's like I haven't changed the settings on anything. Like, I'm just using the same dye for the same stuff. Right, so we've got our little bit of dye there, and now we grab a P1 or P10. We said we're going to do the Keratin this time, so we've got some Keratin. This is making me think that even if this does work, we're not going to get a particularly accurate reading. Somebody says you... Okay. Martin says, wow, you use low binding real brand Eppendorf, you must be fucking rich. They were on Amazon, and they were the only ones that were available on Prime, and said thin wall, and I figured if it works, it's worth it, and otherwise I'll just save them for a rainy day, basically. Alright, let's see if this reads and makes sense. No, that is worse. That is actually worse, and I don't know why. Okay, well, I think this is a wash. Why isn't that working? Do I have... No, I have no ideas. I have no idea. Alright. Well, this looks like it's probably a wash. Okay, well, so we made our calibration chart. Calibration chart worked really good. Then we tried to use the thing to measure some other DNA, and it didn't work. I think what might have happened is I was using just plain water for the well, no, that doesn't make any sense either, because that's also suspended in tea. I don't have a good reason why this isn't working. I'm gonna... Because the thing is, my 0, like this has 0 DNA in it, is reading higher than this one, which has DNA in it. And this was our test sample, so I have no idea. I have no idea why this isn't working. Yeah, this is... Have I mentioned how much I love bio? It's so exciting. So I think what I'm going to do, considering that this is pretty much a wash, and I'm I'm just gonna have to like suck it up and buy the special tubes and the frickin' kit. That's a bummer. Ooh, actually, there's one more test I wanted. There's one more test that I want to run. I did try reading the zero again. Here, look, I'm read. The zero still reads like well over 5,000, whereas the test sample is now reading under 5,000. The calibration tubes are exactly the same. They have not changed. They still work exactly the same. Here's 250. Yeah, the calibration tubes still work perfectly, which means that the amount of DNA in here must be really low or something. I don't know. I don't have any clever ideas. Yeah. Bummer. So I think what I'm going to do is I'm going to probably order the right test kit. And the right tubes, and then this should just work. Like I should just be able to use the regular DNA function. It should just chart this out for me. And you know what? It is annoying that I need to spend more money and buy the kit from the company. But you know what? If that makes this work, so be it. Because I mean, sometimes they're like. The clever workaround does seem to work, in theory. I would just need to... I don't know. Yeah, this is the problem. Should. Like, the calibration curve worked fine. Like, we're definitely getting different amounts of fluorescence at different amounts of DNA. But for whatever reason, both of these test solutions that I just tried didn't work, and I don't know why. In fact, they made the reading go down instead of up, like every other test. I'm gonna... Where's the GFP one? Where's the GFP one? I've let this one sit for a minute. I'm gonna give it another mix. We're gonna test it again, and we're gonna see what we get. Because if the number makes sense, then, you know, wonderful, and it just needed to, like, incubate for a few minutes. But if the number doesn't make sense, then that's a problem. Well, the number makes... still no sense, but it's better than it was before. So, I don't know. Shrug, maybe that means something. Let's try one more time. Once for luck. It shouldn't be contamination. I know there's DNA in those other tubes. Like, I used them recently, and I can see them on a gel, and they all look fine. Wonder if I'm just when I do that shake, it's just to make sure that all the liquid ends up at the bottom of the tube, because that would change the fluorescence. Like if the volume isn't exactly the same, no, this isn't making it make any more sense. It's going down. I have no idea. Well, that's unfortunate. Oh, there was that one other test that I wanted to try. So I'm just reaching up to my incubator. Pull this out. So the one thing I wanted to try was to know if I can use this in a really janky way, like in a way that it was never intended. Which is basically, I want to know if it can tell the difference between... Oh, thank you, Jacob. Much appreciated. Fuck. Where was I? My stuff. Okay. So yeah, the one thing I want to try is I have some very much overgrown... bacterial solutions here, So I want to see if it can tell the difference between like a solution with lots of bacteria in it versus no bacteria. So I'm going to just try... First I'm going to just try pure water. So just absolutely nothing in it, just water. Thank you. And then I'm going to try one with a little bit of bacteria in it. And we're going to see if it can tell the difference. Because if it can tell the difference... Ooh, actually, it just occurred to me that I shouldn't use water. I should use LB for this. Because, of course I should. It's growing in LB. If I didn't, that wouldn't make any sense. I'm going to do a full 200 microliters this time. I'm going to tip this down so you guys can see what I'm doing. So we're just going LB. My LB is over here. Put it over here so you can see it. Cool. So we're going to just pop this into the machine. And we are going to read it. Interesting. Okay, so we get a value of 1856 when reading the green channel. So that's cool. Now let's do the same thing. I'm gonna vortex this tube because there's it'll be hard to see, but there's a pellet of cells at the bottom there. So I'm just gonna vortex these really quick just to resuspend them all. That'll be what that weird vibrating noise is. Don't know how loud that is. That is not. There's still a goober of stuff in that. How's that look? That is not great. Do we have one that's less garbage? One's not grown. That one's not. Yeast. What's this yeast? What is this bacteria? I'm just trying to find something like one of these tubes that suspends nicely. Okay, there we go. We're gonna call that an absorbance of 1. This is my tube over here. Yes, this is overcrowded. Really, I need more space, but I am kind of at a space premium because of the way that we're doing this today. Okay. Now, in theory, there should be stuff in all the various cells that should make them fluoresce a little bit. So in theory, this should give me a different reading. And now when we read it, it does actually. Huh, that's actually really useful. Oh, thank you, Logos. Thank you very much. Greatly appreciated. Okay, so let me just walk you through this again, just so you can see what I just did. So we've got the thing here. I'm gonna do this again just so that I can demo. So this is just a tube with LB in it, so lysogeny broth, nothing fancy. And when we read it, we get a reading of 1844, and that's actually really consistent. It gave me exactly that same number last time, which is great. And now when we read the tube that had the bacteria in it, like lots of bacteria, rendezvous, we get 7366. So it goes up by about 6,000 units, which is really, really good. That's really, really good. In fact, I'm gonna go back to home and I want to try reading this on a different wavelength, just to see what sort of numbers we get. Oh wow, that's fascinating. Huh. Okay, so there's 2 channels in the device, like I said earlier. One of them puts out a blue light and then measures the green light that comes out. The other one puts out a red light, like a red-orange light, and then measures the deep red light that comes out. So when I measured on the deep red setting, The empty tube has a value of 39.8. I'm going to read that again just to make sure that's consistent. Yeah, so 39 point, basically 39, plus or minus a couple of like a percent. Then when we read the other tube that had the bacteria in it, we get 58. So it goes up by 20. So I should be able to now use this every time I want to. So basically what I'm going to do is the next time I've got to do a transformation, so like put some DNA into an organism, before I do anything, I'm going to take a little sample and put it into this machine, and it's going to tell me how much stuff, like it's going to give me a number. And when I do a transformation that works really, really well, whatever number that thing is, that's going to be the number that I shoot for moving forward. Because I'm always using the same LB, I'm always using the same cells, basically. So basically, as long as I get the same number, I'll be like, okay, good. This is it. The cells are at the correct density. Then I'm going to get the special weird tubes and the special weird dye, and then I should be able to I should be able to quantify the DNA concentration. So then so now I've got two pieces of data: how much bacteria I have, or yeast, or whatever, and also how much DNA I have. And so if I have those two numbers, it should mean that my transformations should be much cleaner, because I'll have good data that I can work off of, and then I can translate that into consistent transformations. So that when I go to do something like put two pieces of DNA together and then transform it into a bacteria, which is something I've been doing regularly, I'll be able to have nice, clean transformations every single time, nice and high transformation efficiency, ideally. Yeah, a lot of the stuff that I've been working on lately has really just been going back through all my protocols and really trying to refine them and make them perfect, so that basically so I can stop thinking about them. So like for a while I was having an issue when I was running gels, for example, where all of my gels were just looking like garbage. And I ended up figuring out why, which was I was using water instead of buffer to make my gel. I don't know why. Brain fart kind of thing. But then I fixed it, and then all of my gels have been perfect ever since. So now this is kind of just like, okay, I've ticked off one lab protocol that I can do perfectly every single time. Now I just need to go back through and make sure that I do the same thing with all the rest of my protocols. Because with bio, there's not a lot of different things that you're doing. Like, it's all just mixing small volumes of liquid together. So it's all basically like baking. So I just need to find the right recipe. You know what I mean? Like, I need to find the right amount of flour and the right baking temperature kind of thing. So I'm basically doing the same thing with all of my bio stuff. Like with the spider silk, for example, a lot of the work that I'm doing is just rerunning stuff over and over and over again, trying to figure out like why certain steps didn't work. Which can be very frustrating. It's also why there hasn't been a spider silk update yet. I've mentioned this in previous streams, but there will only be a spider silk update when I have an update. Like, I don't want to do a video about the failure, or not the failure, but like the process. Like, I don't want to just do like piecemeal stuff where, I make a video like, here's another 12 things that I did and none of them worked. Or, maybe one of them worked, but it doesn't necessarily really push the project that much further forward, kind of thing. So I'm kind of just waiting until I finally make some silk before I make that video, is really what's up. And so in the next live lab work, once I've sorted out my camera issues, like I said earlier, we're going to go through and we're going to do just a ton of transformations. And we're going to do a ton of... no, it's mostly transformations. We're going to do a ton of transformations and make sure that we get those protocols perfect. So that way, now we can actually just make sure that that's not a problem anymore. It's not something I even need to think about. It's not something I need to worry about. I can just count on the transformation isn't the problem. You know what I mean? Because when the question becomes, did my reaction work or do my transformations just suck? That's not really a helpful spot to be in, because then you don't know which one it is. So I need to eliminate the doubt about transformation efficiency before I can really zero in on what exactly is the problem. So that's that. But that said, the spider silk thing is coming along quite nicely. Slowly, very slowly, agonizingly slowly, but it is coming along. So as I mentioned before, I figured out the issue, or I think I figured out the issue with why the transformation of taking this DNA and putting it into yeast wasn't working. And so I'm actually going to be sending this DNA to a lab friend who's going to try and put it into some yeast. I'm also going to have the company that made it try and put it into some yeast for me. And so through either of those options, I should end up with silk being produced by yeast. But I hate the species of yeast that this goes into, and I always have, and I didn't want to make the DNA to fit that. It was just what the company had available. So what I'm going to be doing is I'm actually going to be rebuilding this plasmid. So I'm going to be cutting out the silk gene, and I'm going to be putting it into a better plasmid that is easier to work with, has features that I like, and should just work. Whereas this one is just a lot of extra effort. So it's basically a little more effort in the interim. So like it's a little more bench work of, you know, having to reshuffle the genes around a little bit. But after that, it should just be a matter of take the DNA, put it into yeast, call it a day. And if I'm having issues with putting these things together, like the nice thing is there are companies you can just pay and they'll do all of this bench work for you. I want to try and do it myself, A, because I want to film it and so it's more interesting, but also because I want to learn, and also it's just good to have the tools at my disposal, basically. But also, you know, biology is just hard, so we'll see how it goes. If I get too stuck and I get frustrated, I'll just pay a company to do it, because I might as well. Yeah, so that's sort of where that is. I made, and so we'll talk about this on the next Who's Gene stream, which will probably be next week. Friday's stream is going to be, I've got one of my bio friends is going to be a guest on the stream, and we're going to be talking about some of his cool projects. But next week we're going to do another Who's Gene, and on that stream I'm going to talk about some updates I made to the egg and yeast, or the egg and milk yeast plasmids, which have been submitted. They've been sent off to the company. I'm waiting to hear back on a quote. But I made some changes to them so that they're actually going to be going into a different backbone plasmid than the one that I used on the stream. I ended up finding one that was even better, and should be easier to work with and more effective is really, I think, the best way to put it. So those both the egg and milk plasmids have been submitted. Those will get made. Very cool. I'm also going to pay. I'm gonna just pay the company to, once they've made them, just put them into yeast. 100%, whole project finished, ready to go. I don't need to think about it. And it's only an extra like $50 or $60, I think, to have them do that. And I'll pay that. Are you kidding me? Like, not have to mess around with weeks and weeks and weeks of transformations trying to get the thing to work. Yeah, I will pay that. So yeah, that's the thing. That'll be good. I'm excited. I'll show you those plasmids next week. I didn't really make a big change. I just kind of like copied and pasted the chunk out of the old plasmid, stuck it in a new plasmid, called it a day. And then that'll be pretty cool. That'll be done. And then we'll have milk and eggs, and then I can just make some rather than having to like, and then we do the transformation thing. So that'll be fun. Yeah. pretty psyched for that. So I'm working on that. I'm working on the spider silk thing, and that's mainly it. It's basically the majority of the work that I'm doing is just slow. I did a PCR reaction 2 days ago that took 4 or 5 hours to run, and it didn't work. So that's 5 hours worth of running the machine just to check on it later and find out that it didn't work. And so now I get to do that same reaction again. So that's kind of frustrating. Nathan Sandman asks, I hate to be distracting, but did you receive my message about the 2A sequences? Yes, I did, and I changed that in the plasmid. So now it's not 3 P2A in a row. It's now, I threw in a T2A in there as well, so that it mixed them up a little bit. Prithvi Quantum says, no links to papers this month. Oh, yeah, that's a good point. I'll post some. Honestly, so this whole month has been kind of rough. I mean, as I think everybody is aware. Like, people have been asking, like, when's the next video? When's the next video? I had videos planned, and then I had to move my lab and, like, completely change my work habits and schedule, and my access to stuff changed. And so, like, it's taken me a little while to get everything running again. Like, get everything set up, figure out a new schedule, get projects going again. And so, it's just taken some time. So I'm sorry that I haven't been posting links to stuff. I know I should have done that. I'll do that today. I've got loads of papers that I can share, and some of the ones that I'm working off of. The only thing is, like, I haven't read, like, a truly, like, wow paper in a little while. You know what I mean? They've all been kind of, like, most of the stuff that I've been reading has just been really boring, like, baseline papers on, like, transformation efficiency and stuff. It's nothing. I don't think it's something that people would really like. But I do have a pretty good reserve of, like, interesting papers, so I'll be sure to share some of those. Should do COVID-19 research to get more access. I mean, I kind of am. Kind of. I have a positive control, and I mean, I'm working on a video on this. One of the next videos that's gonna come out. So the next video is gonna be lactase. That'll be out in the next day or two, depending on how much editing I get done today. Then one of the next ones is going to be the beta keratin yeast, and then the other one is going to be going through how the testing works for corona. Yeah, so it's going to be a cool video. I have a positive control plasmid. I have the CDC primers. I just ordered LAMP primers, which is another detection method. I've got the antibody kits. I've got basically every detection method that you can buy or make, essentially. I'm just waiting on some supplies for some of them. Like, one of them I'm missing the primers still need to arrive. One of them I'm missing the dye and the enzyme. The other one just doesn't work. So I'm going through and making sure that, like, it's an important video, and I don't want to rush it, and I don't want to put out a half-ass video. Like, I want to make sure that I do it properly and I do it nice, and my information is accurate, especially. It's one of those things that, like, I'm going into it expecting that I'm going to be demonetized, and that's really going to suck. And so that's, like, why doing these streams and, like, people kicking in a few bucks is really nice, because when I make a video that's cost me, frankly, an enormous amount of money that gets demonetized, like, it's going to suck. But in my head, it's worth it, because the content is important, and it's something that people want to know. It's people that I feel that it's stuff that people want to know or people want to understand. And so for me, it's worth it to make that video. So yeah, I'm going to be doing some code. Like, y'all can stop asking. I know you're going to continue asking, but you can stop asking. There will be a COVID video at some point. We're looking like we're going to be in this lockdown for the next 2 years. So it's not like this isn't going to be topical. So yeah, that's going to be the thing. yeah, so that video is coming. I'm really excited to do some testing on it. I think it's going to be really cool, frankly. I think it's going to look really cool. I think it's going to be a chance to explore some really interesting biochemistry, um, and just do some stuff that I don't think is on YouTube. I mean, that's going to be pretty cool, I think. So yeah, that's what's coming up. We got those are the videos that are coming up. There's a few videos that I'd started on that are delayed until after the pandemic, which really sucks because we were making good progress and that just fucking blows, but it's the way that it is. And yeah, so okay, I'm gonna do let's do like five or 10 minutes worth of like question time, and then I'm gonna wrap it up for the day. The next stream will be on Friday, 2 p.m. Same as well, I'd say same as today. Today was late. But yeah, so the next stream is gonna be Friday, 2 p.m. Eastern. We're gonna have a guest on. It's gonna be a lot of fun. And yeah. Then next week we're going to get back to some Who's Gene stuff, and then we're going to also do more live lab work with a better camera. It's probably just going to take me that long to order a new capture card and do the return and all that stuff, which is annoying. What's this digital vaccine thing? I saw somebody ask about. It sounds stupid, but I don't regret asking this. But what is this? Please ban legally bred. Yeah, legally bred, you got one more warning. Like, you've been shitposting constantly. One more and you're banned. Like, it's enough. Are there lockdown laws in Canada? Yes, absolutely. It's, like, real serious. They take it very, very seriously. Everything's locked down, and it's going to be locked down for a long time. Unlike the US, where their response to coronavirus has been approximately the equivalent of, like, in Harry Potter, when, like, the Ministry of Magic was like, Voldemort isn't coming back. Canada is actually taking it seriously and doing a reasonable job of like, trying to protect everybody and provide for people and give good information. So yeah, it's gonna be locked down for a while. What else we got? There was a question about, do you have any good book recommendations? Some, like, we're talking just like fantasy, science fiction, or just like fiction books, then some good ones. The King Killer Chronicles, so The Name of the Wind is the first book. That one's great. Starfish is fantastic. Sphere is good. What other books? What other really good books? Oh, Walk Away. Walk Away is a fantastic book. What else we got? Oh, okay. So the digital vaccine is exactly as stupid as I thought it is. Cool. Okay, what else we got? Do you play any video games? I've been playing The Witcher 3, and League of Legends. That's mostly what I've been playing. Yeah, Walk Away is good, but weird. It's sort of a weird book. I like it. There's a short story. There was a collection of short stories called Everything Change, and in that collection of short stories, one of them is called The Sunshine State. So if you look up Sunshine State, Everything Change, it's a little short story. Highly recommend reading it. It's fantastic. Somebody's asking about glowing mushrooms. Like, what species is the best? Yeah, P. stypticus is pretty great. yeah, it highly recommend. Give us an update about your implanted magnet. I have answered this on literally every single stream. I don't have the magnet. It came out very quickly after that video went out. It never sealed properly. Do you have any future plans to genetically engineer some plants? Yes. We've got a glowing plant thing in the works. We've been working with free genes to get that done, so that's gonna be pretty cool. What is your favorite sci-fi book? It's hard to choose. I don't have one specific book that is like my favorite. I've got some ones that I like. I mean, Starfish I really enjoyed, but yeah, there's lots of great. I love. I mean, I'm a huge sci-fi fan, so most of them are great. Any good books on mushrooms? Paul Stamets's book, I don't remember what it's called, is fantastic. Quick question. How do you have access to lab equipment and resources besides having the right friends? You make a company, like you just make a little LLC or sole proprietorship or whatever, and you find either a commercial postal address or a commercial address, and then they'll just sell you whatever you want because you're a professional. That's it. That's how you get anything you want. Okay, Turdo, you are getting muted because this is just the worst. Why isn't it not? Oh, for fuck's sake! Do I have to do it on this side? How do I make you go away and stay like that? Bye. Anyways, do you know any DIY biologists in Germany? Germany, no. Austria, yes. And their answer to how do you do genetic engineering in Austria is you don't. What they end up doing is they basically they'll design a bunch of DNA and then they'll send it to people in countries that GMO laws aren't ridiculous, and then they just do the they do the work for them. Basically, it's real hard. Doing genetic engineering in the EU is ridiculous because their laws are ridiculous. What else we got? Okay, I'm gonna do one more question. Actually, do you plan to make insulin? So I've been chatting with the Open Insulin guys. I know there's been some many requests to have the Open Insulin people on the show. I'll have one of them on as a guest at some point. They're gonna hopefully send me the plasmids, so I'll have them. But yeah, I'll probably make some for the for the lols, but not like production grade or whatever. What else do we got? Is €125 for a second-hand PCR machine expensive? No, not at all. PCR machines can be very expensive. €125 is nothing. I use the Pro PCR. I really like it. The mini PCR is basically identical. So either one is good. There's that new one that just came out. It's only like $100. It's brand new. For the life of me, I can't remember what it's called. But yeah, there's like a $100 PCR machine now that you can use, like you can buy, and apparently it's great. Why are the EU laws so strict? Is there a sensible reason? There is not a sensible reason. They're just dumb. Like the lawmakers are a million years old and don't know shit about biology, so they just like throw laws at things. It's beyond stupid. Um, Thoughts on the newer bento labs? I mean, I haven't tried them, so I can't say one way or the other. I only ever got to use a prototype model, which I liked in theory. It's just the execution was awful. But it was also a prototype, so I can't say what the final version is actually like. Um, you look tired. I look tired because the lighting is just not great. I mean, the lighting here is not set up for like people face. I thought that I was going to have a camera behind me aimed at the table the entire time. I was not, but then, I realized I'm an idiot and I bought the wrong capture card. So, uh, that's why I look so tired. I'm not actually tired. I slept really well last night, surprisingly. Um, what's your take on DNA is not a blueprint from the Scientific American article? I haven't seen that article, so I don't know. Shrug. Um, have you thought of using yeast to produce CBD? No, because I have a friend who's already done it, and it cost them millions of dollars. So, I don't care. Also, there's a perfectly good plant that does that just fine. Um, okay, well, let's come on. I want one good question to wrap this up, and then we're gonna call it for the day. Anything interesting? Any Hawks experiments to do at home? I don't mess with Hawks genes. That's getting into like playing with fruit flies or something, and I'm like not into it. Um, Also, I just don't know enough about Hawks genes. They're so weird, and I haven't read up on them enough because it's not something I tend to mess with. Um, uh, mm, how close are you to real spiderweb fluid? That depends. It could be 2 weeks. It could be 2 years. It depends when my bio stuff starts cooperating. Um, uh, um, let's see. What is your take on more high-end analysis techniques like mass spec, NMR in bio? Um, I mean, you don't really like mass spec and NMR. Like these tools are used in bio, but only for very specific things. Like it's not like the reason everybody uses gels or whatever or a qubit, for example, is because those are the things that you need when you're doing bio. Like you don't need a mass spec for doing bio unless you're trying to classify a protein or a weird compound that a weird like creature makes. And you do use a mass spec for those things. It's just you don't. It's not the sort of thing I would use every single day. Um, all right, you're getting timed out. I do not care about your your uranium. That just no. um. I well, it's 4:30, so we're probably gonna call it there. Actually, there's one quick one. Do you need a laminar flow hood to grow mushrooms? No, I've done it without. As long as you make sure to really like autoclave the crap out of your stuff and just work cleanly, you can totally do mushrooms without or grow mushrooms without a flow hood. Flow hood makes it nice. Then you can be a lot lazier, but it would be help. You can do it without. All right, cool. We're gonna call that for the day. I hope you guys have enjoyed. I know this has been a little weird, and it's a new format. We're still learning. The next streams are hopefully gonna be in higher quality. We're gonna have more camera angles. We're gonna have all kinds of interesting stuff. We're gonna have guests on coming up. We're gonna have all kinds of stuff. If you haven't already, there's a new video up on my second channel, which you should definitely go check out. Look up the Taste Emporium. Made some sourdough, which was real fun. I hope everyone's going to stay safe and have a wonderful, wonderful rest of the day. You know, don't get sick. Don't lick doorknobs. Have a good day. Try not to get too frustrated. And, yeah, we'll see you Friday.